With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hi students, the question given is, in the lac operon, the structural genes are switched off when? Our options are repressor binds to operator, to promoter, to regulator, and to allolactose. So lac operon is used in the process of transcription, and when the structural genes which help in the transcription process, when these structural genes, they are switched off, so transcription is hampered. So if we see our options one by one, the first option given is repressor binds to operator. So when repressor binds to operator, this operator is a specific DNA sequence which is present in between the promoter region. This promoter region, it promotes transcription, so it is present in between the promoter and the first coding gene. So both these two, they help in transcription, but these DNA sequence which acts as an operator, it is present between the promoter and the first coding gene, and that is why what happens, the repressor, here the repressor protein binding site, it serves this DNA, then it serves as a repressor protein binding site, and thus it prevents the RNA polymerase at the promoter site. So what happens here, since this DNA segment, it prevents the RNA polymerase to go to the promoter site for transcription to occur. Thus, this DNA segment, it switches off the structural genes as it is present in between the promoter and the first coding gene. So this region then becomes a repressor protein binding site. I'm writing it down for you. This region becomes a repressor protein binding site, and thus the structural genes are switched off. So this first option can be the right option for our question, but let us see the other options. The second option is repressor binds to promoter. So this promoter is the region which facilitates transcription. So this is a DNA segment, this promoter, and this DNA segment, it helps to initiate transcription of a specific or a particular gene. So a particular gene's transcription occurs because of this promoter, and structural genes are activated. So this is a wrong option. What happens when repressor binds to regulator? When repressor binds to regulator, these regulators, they are genes that regulate expression of the structural genes. So these regulators, they have protein products. So here the genes, they are not switched off, but here there is structural alteration of these genes when the repressor binds to the regulator, because it has proteins which look like the promoter site, so there is structural alteration, but the genes, they are not switched off. So this is also a wrong option. And lastly, when repressor binds to allolactose, so this allolactose is, again, this is an inducer, so this helps to induce the transcription process, and thus it is an inducer in lac operon, so it induces the lac operon, it helps the lac operon to function and promotes transcription. So these are the wrong options here. These two, promoter and allolactose, they promote transcription, and regulator, here they altered the structural gene, but the structural gene is only switched off when the repressor binds to the operator site. So this is the right option for our question. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.